Okay, so just a brief introduction for everyone. So Lisa is professor of physics at Syracuse University. She did her PhD at University of Santa Barbara in California and then moved to Princeton for an independent postdoc at the Princeton Center for Theoretical Science. Lisa started her own group in 2011 at Syracuse University, and since then she has been studying collective behavior in biological tissues and deformation in inert materials. She uses computational and theoretical techniques to understand and predict emergent behaviors in tissues, and in particular the transitions between different tissue states from fluid to solid and vice versa. She is working with many different models, including zebrafish morphogenesis and cancer invasion, and she has many fruitful collaboration with a number of experimentalists. So now, thank you for accepting our invitation, Lisa, and for presenting your work on predicting mechanics of 3D epithelia in vertebrate embryonic development. And without further ado, please, thank you very much.
Thanks so much for the nice introduction and for the invitation. Hi, everyone. Thanks for being here. The first thing I want to say is that I would be very happy to be interrupted during this talk. So my background is a physicist. I work a lot with biologists, but if I say something and it's not something you're familiar with or you have a question about it, I'm sure everyone else has the same question, so please interrupt. The next thing I'll do is highlight the people who actually did the work. So this is my research group, and in particular today, I'm going to be highlighting some work done by Raj Kumar Mana. I'll highlight people when they show up. Liz Lawson-Keister, Somielo Azote in my group, and then I will be sharing some data and work from a collaboration, a really fun collaboration with Corinne Neeson and Sarah Wickstrom, and also in a collaboration, a separate collaboration, and that's in mouse, and then a separate collaboration in zebrafish with Jeff Amack and Heidi Henley's groups here in Syracuse. Okay, and so I have two stories to tell, and we'll see if I get through both of them today. I might only get mostly through, one and have just a little bit of time for the other. But the goal is to understand the mechanical environment and how the mechanical environment impacts development in epithelia. And in particular, I'm interested in doing that in sort of complex 3D environment. So either where you have like a stratified epithelium, so the epithelium itself is sort of multiple layers, or in the case of the left-right organizer in zebrafish, you have basically a monolayer epithelium, but it's in a spherical shape and it's embedded in a three-dimensional tissue. And I'm interested in how those interactions work. And the goal is to understand which mechanisms are driving tissue morphology changes. And I really mean mechanisms in the molecular mechanism sense. At the end of the day, that's where we want to end up understanding. Okay, but in order to tell you those two stories, I first need to tell you a story of we also need to understand how forces are transmitted through tissues, because if I'm interested in how mechanics of tissue is impacting the shape of epithelia, I need to understand if there are forces being generated by convergent extension or other things, how those forces are transmitted through a three-dimensional tissue. Okay, and the question, the way that you may think about, like if you're thinking about, okay, forces being transmitted, there's a big difference between depending on sort of the material properties of the tissue. So one way of asking the question is, if I have a force over here, is the force, if I push over here, is the force that I feel over here dependent on how fast I'm pushing on the tissue or how far I've pushed the tissue? Because that's going to result in very different tissue morphologies in response to that mechanics. And so why do I say those two words, fast or far? Well, I want to give you an example of one type of material that, of course, you're all familiar with, which is water. Okay, and so if I take a glass of water, it pours, which means that it sort of changes its shape very easily. And then if you want to think about how forces are transmitted, you can ask yourself, well, if you're in a pool, is it easier to run or to walk? And of course, it's easier to walk through a pool. If you try to run through a pool, it's really hard. And why is that? It's because for all liquids, the way that forces are transmitted, it's dependent on how fast you're moving. So how hard you have to push on water depends on how fast you try to move. And when you push, the material doesn't return to where it started. And if you want to understand why liquids behave that way, you can understand from a microscopic perspective. That's because the constituent particles, in this case molecules, that make up this material, they're always rearranging and changing their neighbors. So that means there's no sort of reference state that you can think about. And that's why it doesn't matter how far you push it, it just matters how fast. Okay. In contrast, if I have an elastic material, and I think the canonical example that we're all familiar with there is a rubber band, is that you can stretch it and it's elastic, okay? And so what happens is you stretch it, well, you all know it gets harder and harder to pull a rubber band the further you stretch it, right? So it's a solid, and that means the force is higher as you move farther, right? How fast you move doesn't matter, it matters how far. And it does return to where it started. So you stretch it, but then you let it go and it goes back exactly the way it was. And that's, again, at the microscopic level, because the constituent particles are not changing their neighbors. Okay, and so if we want to understand how forces are transmitted through tissues, we want to ask the same set of questions. Are tissues solid-like or fluid-like? And importantly, because we care in the end about mechanism, how do cells control these tissue scale properties? So the first point is that these emergent tissue scale properties are really important for morphogenesis, and we'd like to understand how cells can control them in order to impact, for example, epithelial morphology. Okay, so the first thing that you probably are possibly familiar with is that in the past 5 years or so, there's been a lot of new papers out showing that in developmental systems, tissues are constantly changing their fluidity. So they're actually transitioning all the time from solid to fluid. So this was a paper by one of your colleagues about the fluid solid transition across the zebrafish embryo. And so the idea is I hate talking about other people's work in front of them. But anyway, as you go towards the head of the zebrafish, there's a more solid-like behavior to the tissue. And then as you go towards the tail in this tail bud, the tissue is more fluid-like. And there's some really beautiful work to characterize that carefully. And then more recently, there was another, again, this is still in zebrafish, but a very different stage. It's at the dome stage where the cells are just sitting there on top of the yolk in the zebrafish. And there's this process where the tissue starts to flow over the sides of the yolk. And that's basically the initial elongation of the body axis. It's a little more complicated than that, but basically there's this big, large scale flow of the tissue. And so if you think about it, right, if I had a solid-like tissue, if this dome was solid-like, and I had to stretch it out this far, because if it were solid, that would cost a huge amount of energy because I have to stretch it really far. But if there's a transition where the system transitions to being fluid-like at exactly the right time, then this large-scale flow wouldn't cost any energy at all, or very little energy. And so actually there's some careful work showing that's exactly what the biology does. It actually tunes itself that right when that large-scale deformation happens, all of a sudden the system loses rigidity, it becomes fluid-like, and that enables the morphology change. Okay. And so another key point is that it appears that biology can use multiple mechanisms to generate these large-scale changes in the tissue mechanics. So one of the things that you can do is you can change the level of fluctuations in the system. So if this were like an inert material, this fluctuations axis can be thought of as something like the temperature, right? If I give you a glass of water and I tell you to make it a solid, you just stick it in a freezer. And that's because you're actually reducing the amount of fluctuations. And we all know that if you reduce the amount of fluctuations, you can generally make a system more rigid. Then there's another axis, which I've called here in this diagram 1 over the density, right? And that also makes sense for sort of standard physical materials. So you may also remember that if I told you could not use a freezer and you still needed to make a glass of water a solid, the other thing you could do is you could compress, the water, because it turns out that, pressure is also a good way to solidify a material, or changing the packing fraction or the density of the material inside. Crowding is another word that people use. So compressing a material is another way to make it a solid. And then it turns out that there is a third axis, which is often used by biological systems. And this one is a little less intuitive. And you can tell it's less intuitive because it has a horrible title over here. It's called geometric incompatibility. But it turns out it's one that's really important for epithelial layers. And that's because in an epithelial layer, all the cells are touching all the other cells already, right? All of the cells are sort of, because epithelia need to form a barrier. And so all the cells are touching all the other cells. And so in that case, you're already at a density, you're already at a packing fraction 1, you're already at the biggest density you can have because you're already taking up all the space in the system. And so if you want to transition between solid and fluid, you need another mechanism. So because it's something that we've in my group have worked on a lot, and also because it's really important for epithelial layers, I'll just take a moment to talk about it a little bit. Maybe one thing I'll say is that in these two systems that I sort of talked about already, it turns out that crowding or jamming or changing the density and changing the fluctuations, those two things together, are really what is causing the rigidity transitions in these two cases. So these are not epithelial-like systems. And in these cases, the data really shows that you go along one of these axes in this diagram. You go sort of along the density and fluctuations axes. But in epithelia, you can't do that. Okay, and so what happens in those cases? So I'm going to introduce the model now because I'm going to also use the model for later in the talk. If you imagine an epithelial monolayer, and here's like a very schematic diagram of an epithelial monolayer, you have, adherens junctions and an apical ring of actin and myosin. They're attached to a basement membrane, and there's a lot of, depending on exactly how stiff that substrate is, adhesion with the substrate, usually via integrins and stress fibers down there. But if I take a bird's eye view and I kind of squint my eyes a little bit, basically an epithelial monolayer looks like a packing of polygons. Okay, and so here's a picture of, a close-up of a few of those polygons. And if you want to make a simple model that's probably wrong and you don't expect it to be right, but anyway, you can ask, well, what are the geometric features that I could try to write down in a mathematical model for the system? And so, obviously, if you're looking at a bird's eye view, there's the apical area, there's the cross-sectional area that you see, and then each cell also has a perimeter that you can define for this two-dimensional view. Okay, this is, I think, the only equation in the talk. Basically, what it says, there's two ways to interpret it. There's a physicsy way and I think a more biology way. So I'll do the physicsy way first. So the physicsy way is you sort of assume that whatever cells are doing, they have a homeostatic cross-sectional area that they want to maintain. So I'll call that A naught. And via all of these mechanisms in signaling, they also maintain a homeostatic perimeter. And maybe those two things are kind of independent. Maybe they can regulate those two things independently. So the cross-sectional perimeter that cells want to have, I'll denote P0. So that's the zeros mean that's what the cells want to have. And then I say, well, if the cells want to be at this area and this perimeter, then they should pay a penalty whenever they deviate from that. So I have the actual area minus the target area. If I sort of square that, I say, okay, there's an energy for deviations, either positive or negative away from that. And so this is the simplest mathematical model you can write down that has that feature, okay? And similarly, I just say, okay, I pay a penalty for deviations away from my target perimeter too. Okay, but that's sort of, okay, like really what's going on under the hood. Well, there's been a lot of work suggesting that actually, so I can multiply this term out. And if I have a term that's proportional to the perimeter in 2D. That's basically a tension-like parameter, like a surface tension or an interfacial tension between the two cells, because it's proportional to the perimeter in 2D or the surface area in 3D. So terms that are linear in P look like an interfacial tension. And so that's going to be regulated by usually cadherin-based adhesion and cortical tension in the cells. There are a couple of different origins of a quadratic term in P. All that means is like I start to pay a bigger penalty the bigger my perimeter gets. So one way you could get that is if I have an actual contractile ring of actin and myosin that's trying to contract. Another way you can get a term like that is actually if you have a limited pool of adhesion molecules or actin and myosin that can be promoted to the boundary of the cell. And then a way that you can get a penalty on area fluctuations, which is really natural, is actually most of the cell types we study are almost incompressible. So the ion channels aren't open and they're not changing their volumes. And so then if I have a monolayer and there's resistance to height fluctuation, so again, I'm sort of looking at now a lateral view again. So there's sort of the cell, like one cell right next to another cell, they can't be vastly different heights. Well, if they're all about the same volume and they don't change their volumes, then that actually gives rise a sort of barrier to height fluctuations, or an energy penalty for height fluctuations actually gives rise to an energy penalty for area fluctuations too, because the volume is just the product of the height times the area. Okay, so that is how you might understand the origin of this model. This is called a 2D vertex model. But it makes a it's a stupid model. So it's a very stupid model, but it makes a set of striking predictions that you can then go and test and experiment if you're brave and willing to believe stupid models. Okay, so what is the prediction? Well, there's immediately one parameter that jumps out at this equation. So if you do a non-dimensionalization of this equation, there's one parameter that jumps out, and I'll call it the target shape index. And it just jumps out of the math because it's the dimensionless parameter inside these equations. It basically says that if I look at the ratio between the perimeter of a cell to its area, then if I have a very small perimeter relative to my area, I'm going to be compact. I'm going to be sort of rounded up. So small perimeter relative to area is compact. And then as I make the perimeter larger, as this target shape index, which is the perimeter over the square root of the area, as that gets larger, I get more and more like elongated. Okay, that's the idea. And so what we see in simulations then, and also in some analytic theory, is that actually there's a prediction that the system changes from a fluid-like state to a solid-like state at a specific value of this shape parameter. Okay, and I'm going to show you some data in a second. It's from fruit fly embryos. So this was some work in collaboration with Karen Keisha's group at Columbia. But before I go to that slide, I also want to tell you, in fruit flies, there's this additional thing that's going on, which is, I'm sure you all know, that there's planar cell polarity that generates anisotropic tensions. So all of this that I'm showing you here is in an isotropic case, where there's no shear forces from outside and there's no planar cell polarity. But if I turn that on, I also need to think about what happens when I have sort of more stress coming from one direction than the other. So it turns out that we've done a lot of work in order to make this theory handle that case. And so in that case, we don't need just this cell shape index. We need another term, which is the cell shape alignment, which is how the long axis of each cell is basically oriented relative to another. So if the cell axes are highly aligned, that's a high cell shape alignment. And if they're not aligned, like I have one this way and one this way, that's a low cell shape alignment. Okay, so this is a very busy plot, but I'll just walk you through it because I think it's one of the sort of the evidence that this type of rigidity transition happens in real epithelial layers. Okay, so here on this axis is this shape index. And on this axis is that cell shape alignment that I told you about, whether the long axes are aligned or not. And this black line here is a theoretical prediction a priori from the theory with no fit parameters. So I just want to emphasize that again. I have not taken this data and then shifted this curve around. This black line is no fit parameter. It is a prediction of the theory, okay? So we can't change it. It's fixed. Okay, and if you want to look on maybe along this axis, if there's no shape alignment, so this shape alignment is 0, then what this says is that if I'm below a shape index, so I'm a more rounded up cell, so if I'm below a shape index of about 3.81 or 3.82, then I expect to be a solid. Okay, that's what this black prediction is. It's a line that separates solid-like behavior from fluid-like behavior in the theory. And so if I'm below this line, I'm a solid. And if I'm above this line, the theory predicts that I'll be fluid-like. And again, this will change how forces are transmitted through the tissue. So then the data that you see on top of this curve is taken from body axis elongation in Drosophila. And so what we do is we segment the whole tissue and we can explicitly quantify these two, because these are just numbers. So once I have a picture of a tissue, I get this number and I get this number. I get the shape alignment and the shape index just from looking at a picture of a tissue that I've segmented. Okay, but then, of course, because it's Drosophila, we can do additional things, which is we can count the number of rearrangements per unit time in an embryo. And so this, I don't know if you can probably see it on your screens. I know Zoom is hard, but this upper corner is this inset is the trajectory of what happens for a single embryo. And so what you can see basically is that over time, the system basically starts here and then it moves out and then it comes back. Okay, and what we're doing in the color, so this color bar is it's the number of rearrangements that we see in the tissue per unit time. So it's the rate of cell rearrangements. Okay, and again, we expect that if we're in a solid-like tissue, there's essentially no rearrangements. And on this color bar, that's the blue colors, are basically no rearrangements to within our experimental imaging error, okay, is blue. All the other colors are basically rearrangements are happening. We can see that there's rearrangements happening. And so what you see is that basically for this one embryo, when we cross this line, that's the threshold of where the theory predicts solid and fluid, you see all of a sudden you go from no rearrangements to lots of rearrangements. Okay. And then what you're seeing in this big picture here is lots of embryos. So this is data averaged over lots of embryos. And again, what I think you can see pretty clearly is this black line does a remarkably good job of separating solid-like behavior where there's no rearrangements in the Drosophila, the blue, from the red and yellow colors, which is where there's lots of rearrangements. Okay, so the idea here is that you can take a snapshot of a tissue and you can predict which state the tissue is in, solid-like or fluid-like, if it's in epithelia and epithelium. And you can then think that gives you an idea that both this vertex model at least is capturing this transition remarkably well and quantitatively. And then it might help you understand how forces are transmitted when you want to understand morphogenesis. So we're going to take this now as a starting point for the rest of the stories. Okay, maybe I'll ask, are there any questions? Because I'm about to launch into like specific stories. Okay. Zoom is so hard. I feel like I'm shouting into the void, but I hope you're all doing okay and not too bored. Okay, so so the current work we're interested in the left-right organizer in zebrafish and the mechanics of the mouse stratified epithelium. Okay, so the first story is in zebrafish, and a lot of these are really extensive set of experiments and the image analysis. So a lot of people have contributed. Let me make sure you're familiar with the system. So this is a zebrafish body sitting over a yolk, and the left-right organizer that determines left-right symmetry breaking in zebrafish is called Kupfer's vesicle. So I'll call it KV throughout the talk. And it basically is a transient organ that sits here in the tail bud of the organism. And so here's a different view. And the key thing is that this is the view I'm going to show you for most of the rest of the talk, where the anterior, the head of the animal is up, the bottom of the tail bud is down, posterior, and then left and right, okay? And dorsal ventral is in and out of the page. Okay, so what my collaborator Jeff Amack has shown is that there's a particular set of cell shape changes that are upstream of all left-right patterning in the system. And it's a weird thing. It's basically that in this, it's shown in panel B, when this organ forms, it's totally symmetric anterior to posterior. But from the 2 somite stage to the 8 somite stage in the animal, the cell shapes become very asymmetric. The cells on the anterior of this epithelial layer that surrounds the lumen, they become very elongated. And the cells on the bottom, or posterior side of this organ, they become sort of very short and squat. Okay, so they become sort of elongated in the opposite direction. Okay, so short and squat down here, long and skinny up here. Okay, and we're going to use this as, and I maybe should say that there is similar ciliated organs in chick, mouse, and human. So we're going to view this as a canonical example of like, okay, this is like a very simple organ that's forming, and we want to understand what's driving these shape changes, these cell shape changes that are important for the function of this organ. That is our goal. And I've been working with Jeff on this for a long time now. Okay, so, maybe I should also say, why do we think this shape change is important? So this lumen inside this organ so each one of these cells is monociliated. And basically the cilia are also tilted, and they're all tilted in the same way. And when they start to move, they generate a counterclockwise fluid flow inside the lumen. And it turns out that, the cells, although I say the system is kind of weirdly understudied, but anyway, there's a set of cells over here that are on the left-hand anterior and slightly dorsal side of this organ that read out the shear stresses that are generated by this fluid flow. Okay, this is a crazy system. Okay, but it's what happens apparently. Okay, and so our hypothesis is that because the readout is up here on this sort of left-hand side and the flow is counterclockwise, we actually, and the cells are monociliated, we actually need to pack a lot of cilia on this anterior side in order to get a strong enough flow that it triggers these stress sensors. In other words, we think that if you don't pack enough cilia on the top, then you don't get a strong enough amplification of the signal and readout later downstream. Okay, I mean, I would say the evidence for that is not extremely strong, but actually there's all of this doesn't have a ton of evidence. It's still kind of a mystery. Okay, but what is true is my collaborator has shown that if you block these shape changes, left-right signal or left-right patterning happens incorrectly in these organisms. So this shape change is clearly upstream of this left-right symmetry breaking. Okay, and so for the rest of this talk, I will talk about an important observable in both experiments and in modeling, which is the number of cilia that are on the anterior side, right? Because if I make all these cells on the top long and skinny, then I can pack a lot more cilia because they're all monociliated. So we'll count the number of cilia on the top and the bottom. And here's an image where we've labeled the cilia, so you can see it's something we can do pretty easily in an experiment, which is sort of, and you can see in this picture that there's a lot more cilia on the anterior side than the posterior side of this organ. Typically, this ratio in experiments is somewhere between 1.7 and 2. So you actually pack almost twice as many cilia on the top as on the bottom of this organ via these shape changes. Okay, so of course the question is what causes these cells to change shape? Okay, and so when I very first started my faculty position at Syracuse, I said, well, maybe there's some upstream morphogen gradient, of course, that generates differential. So that just says, okay, these anterior cells have different mechanical properties than the bottom, and there's a morphogen gradient. And so depending on the level of that morphogen gradient, there's more, these cells are different. They're different than the cells down here. And of course, that will generate the shape changes. So this is a model showing, yeah, that will generate the shape changes. But the problem is that Jeff Amack, and lots of other folks in the field have been looking for a morphogen gradient in the system for 10, 20 years, and none has been found. And we've looked at all of the, it's difficult to say we will never find one, But like we've looked at all of the obvious ones, we think, and no one has ever reported a morphogen gradient that's acting in this way in this system, and we can't find it either. Okay. So another possibility is like based on this paper that says there's a static gradient in tissue fluidity. What I mean is that the paper by Ale and others basically says that there is more fluid-like tissue in the bottom and in the bottom of the tail bud, sorry, and more solid-like tissue on top. So I call that a static gradient in the sense that like the system sets up that gradient in mechanics, okay? And so we're like, well, maybe the KV takes advantage of that gradient in mechanics to change these cell shapes. So we looked at that very carefully in vertex models. And the bottom line is it helps a little. This static gradient can help a little to generate those shape changes, but it really cannot explain them. In other words, it's just not sufficient to pack a lot more cilia on top. It doesn't work. And we've tested that in the, like, it just doesn't, it's very hard to make that mechanism do that. Okay. So then, of course, I'm going to now show you a movie of the, this organ as it's forming. And so in particular, I'm going to show you a movie from the 2 somite stage to the 8 somite stage, which is precisely when these cell shape changes are occurring. So this is the Kupfer's vesicle is labeled in green in this movie. And then you can also see some labeled nuclei that are the darker gray of the surrounding tail bud tissue. And actually, if you look, you can kind of see the notochord, which is sitting here on top of Kupfer's vesicle as well. Okay, and it's undergoing convergent extension. So what's happening here is sort of the notochord is undergoing convergent extension and it sits on top of the Kupfer's vesicle. And our assumption is the convergent extension in the notochord is helping to push Kupfer's vesicle downward through the tail bud tissue. So let me play that movie again. I think you can see it. Okay, so you can see like the cavia is moving and also the tail bud is moving. But what's, I think, very cool is if you basically change this movie, so you keep Kupfer's vesicle as the reference frame. So you sort of, you say, okay, I'm going to make Kupfer's vesicle not move, and I'm going to look at the motion of the tissue around it during this developmental process. I think hopefully you can see this gorgeous picture of basically the cells moving around this organ. Maybe I'll play it one more time. I think it's really striking. So I'll play it. I mean, okay, I'm a physicist, so I like it. But anyway, you can basically see that as a function of time, like this organ is like basically has this like flow of cells around it effectively. Okay, and so of course, that makes me think of, and oh, you can quantify this flow very carefully using particle image velocimetry. And you can extract the streamlines, which are basically the lines of constant velocity around this organ during the relevant developmental time period. And this reminds you, and this is a picture from a classic book of fluid dynamics pictures of what's called Stokes flow around the sphere. So the idea is that even if you're at very low Reynolds number, which means flows are very slow, you basically have, if you have a spherical object and you flow around it, the flow has to bend, and that generates drag forces on the sphere. So you basically get a pushing force sort of at the front of this, well, here, you know, at the beginning of this flow, at the bottom of the sphere in this image, you get a smooshing force, if you will, a smooshing drag force. And then at the back of this object, you actually get a drag force that tends to elongate things at the back. So it sort of smooshes at the front and elongates in the back. Okay, and that's a little bit interesting because, of course, that is very much what sort of the shape changes of the cells in this organ kind of look like. Okay, but you can't just say things look like one another. That's not a very good thing to do. So we need to understand whether there are drag forces and whether the magnitude of those drag forces are sufficient to cause shape changes in this organ and in these cells. Okay, so the other point I want to make to this audience is usually these types of forces are ignored in development. Actually, I ignore them when I do work on Drosophila, or when I've done all of the previous modeling work in developing systems. I have ignored these dynamic drag forces. Okay, and why have I ignored them? And I think everyone else in the field has mostly ignored them, is because the motion is very, very slow, right? This organ moves about 1 micron per minute, right? Okay, and so if you have fluid, so again, that we think that perhaps from previous work that the behavior of this tissue around it is fluid-like, so it matters how fast I move. Okay, but the forces will only be large if I'm either moving very fast, but I'm clearly not, or if the viscosity is enormous. And so viscosity is something you're probably familiar with, right? Like honey is way more viscous than water, and it's much harder, it's more difficult to pour because it's more viscous. And so the forces in this system, because the motion is so slow, the forces will only be large if the viscosity is absolutely enormous. But the cool thing is that previous work has shown that these tissues are near a fluid solid transition in the tail bud, right? And so that means that when you get to be a solid, the viscosity diverges, it goes to infinity. Okay, so that means that's really exciting because it means the viscosities could potentially be quite enormous inside of tissue. So that means slow motion could generate large drag forces. So perhaps we should not be ignoring it. Okay, but in order to test that quantitatively, we need a mechanical model for cell shapes and tissue fluidity in 3 dimensions. Okay, so very briefly, I'm going to go through this very briefly. I lied. Here is another equation, but this equation is exactly the same equation, just in 3D. So now instead of perimeters, so that was the lines in 2D, I have surface areas in 3D. So I have a target surface area now, and that takes the place of perimeter in 2D. And then instead of areas, I now have volumes. But otherwise, this is exactly the same equation as before, just generalized to 3 dimensions. And it turns out that there's exactly the same parameter, except to non-dimensionalize the shape, it's the surface area divided by the volume to the 2/3 instead of the square root, just to get a dimensionless shape index. But it turns out that exactly the same thing happens, where if you have a compact shape in 3D, then the system is solid-like. And as you get less compact, more surface area to volume, then the tissue sort of collectively becomes fluid-like. Okay, so this is just some data. And in 3D, that number at where the transition happens, it's not 3.81 anymore. It's about 5.4 in these with this new definition. Okay. Another thing that we want to account for, and this isn't an important detail, but just so you've seen it, in this situation where we have Kupfer's vesicle inside a tail bud, we clearly have different tissue types that don't mix with each other. And in general, in models and actually also in biological systems, the hypothesis for why they don't mix with one another is because there are heterotypic interfacial tensions generated by signaling cascades, many candidate. I won't get into the biology. I'm sorry, I won't get into the biology of this today. But heterotypic interfacial tensions, we need to put those in our model so that like the cells that we define as Kupfer's vesicle, epithelial cells, do not mix with the surrounding tail bud tissue. Okay. And the last thing I need to say, and this is a technical detail for experts, so I won't belabor the point, is that actually, we can't get away with a Voronoi model, which is what we had been using in 3D. It's a little bit of a simpler model to compute. Computationally, we need a vertex model. So for this audience, I'm not going to say anything else, but if you care about this, I'm happy to talk about it more. And I need to highlight that we use some open source code for 3D vertex models that was developed by Tao Zhang and Jen Schwartz. And so if you are interested in doing modeling or you have collaborators that are, I think it's a real service to the community that Zhang has made the code available open source. Okay, so Raj, who's a fantastic postdoctoral associate in my group, has basically made this 3-dimensional vertex model for the Kupfer's vesicle moving through the surrounding tail bud tissue. And in our model, it's just because it's easier to handle computationally. We also have the lumen made up of like cellular structures. This area is filled with fluid in the experiments, but we checked that the parameters we choose for this fluid don't matter so much. In other words, although I represent them as cells, this works, that this is basically a nice representation of the lumen. Okay, and so now we can do our sort of experiment well, a numerical experiment, a numerical simulation, where we say, okay, what happens if I just go in by hand and I drag this Cooper's vesicle, which I now model with these 3D vertex models, which we think are really reasonable representations of the mechanics of how cells interact, especially in these systems that are very close to a fluid-solid transition. So we just drag it through the tissue and see what happens. So this is a video of the simulation, and it's a periodic boundary condition, again, for computational reasons. But hopefully, if I pause it right here, you can see that basically it's cool. The simple act of dragging this object through the tissue does in fact generate precisely the cell shape changes that we see in experiment. So here I'm reading out the number of anterior cilia divided by the number of posterior cilia. And you can see you basically get something that's really in the physiological range of about 2, that ratio being about 2. And you can go in and see a comparison with, so here, I'm sorry, I'm showing you this right here. So this is what the data looks like if we look at a similar view inside the experiment at the 8 somite stage at the end of this process. Okay. So the other thing is that the nice thing about simulations is we can ask, okay, where in face and parameter space of this vertex model does these shape changes happen? And so this is the amount of basically force that we apply to the KV to drag it through the tissue. And you can see, well, as we increase the amount of forces, that actually, of course, makes the thing move faster. And that means that I typically get more drag forces. And the other nice thing is that I only get those drag forces when the system is fluid-like. So this axis here is how fluid-like is the tissue. And you see, I told you that there was a transition from solid to fluid around 5.4. And you can see that once I get into the fluid phase, that's when I start to see these shape changes. So it's really a feature of dragging a system through a high viscosity fluid, as we anticipated, that gives these shape changes in the model. Okay, but I already told you the problem is that the motion of the KV is very slow. And so we're still left with the problem, are drag forces large enough? And that's because, quite frankly, it's still very difficult. We have some collaborations who are trying to do this, but we can't go in and measure this S0 parameter in an experiment, because this is the shape that cells want to have. And so it's not something that it's easy to go in and measure in an experiment. So we had the idea alternatively is as well, okay, our prediction is very clear that this effect depends on the velocity of the KV through the tissue, and that if the velocity is lower, this effect should be a lot lower. The shape change should be a lot lower. Okay, and so the question we were after is, can we perturb the velocity of this Cooper's vesicle without changing anything else? And the answer is, we thought, well, because convergent extension in the notochord, we think, is driving this motion. So now I'm showing you basically, I'll show you a picture of where we'll go in with laser ablation. And this is a gigantic, this isn't some little precise laser ablation. We're going in and trying basically to disrupt the convergent extension in the notochord. So we are careful not to do it close to the KV, because we want to keep the portion of the notochord that's touching the KV intact, but we'll ablate it above the KV. And the hope is that by ablating a chunk of the notochord up here, we're not perturbing the rest of the system, other than we're slowing down the rate at which this Cooper's vesicle is moving through the tissue. So we've done a lot. These are incredibly hard experiments, incredibly difficult. And what we notice is that the laser ablation of the notochord doesn't stop the motion of Cooper's vesicle. So here's Cooper's vesicle down here. Here's the notochord. You can see that there's still some net motion towards the posterior. But the cool thing is that we clearly slow it down, and we slow it down by a factor of 2. So this is basically the control system and the ablated system. And you can see that at the end of this experiment, we've basically slowed down the motion of the system by about a factor of 2. Okay. And so it turns out that in these experiments, it's because of the restrictions on the Zoom, basically the objective we can use to do it, it's hard for us to characterize individual cell shapes, but we realize that we can characterize the lumen shape, the shape of the lumen very well. So here is a typical image from one of these experiments where we've done a laser ablation, and you can see that you can pretty easily characterize the shape of the lumen, although characterizing cell shapes would be more difficult. And so what we're going to do is analyze lots of ablated embryos, and we're going to characterize the shape of the lumen. So here's a bunch of control embryos. What happens to the lumen shape? And here's a bunch of ablated embryos. And actually, we have more data than this. This is actually now a subset. Okay, I guess I can't skip this entirely. How do we characterize the shape of the lumen? There's lots of ways we could do it. What we've decided to do is characterize the radius of gyration of the shape of the lumen. So if I have an object which is circular, and I take the like the sort of average displacement in the x direction relative to the x direction relative to the y direction of that shape. If I have a shape that's spherical, the ratio of those sort of 2 radii is 1. If I have a shape that's elongated like this blue one, then I have the radius in the y direction is less than the radius in the x direction, and vice versa. If I'm elongated in this direction, then I have this number being greater than 1. And so I guess this is I'm almost out of time. So clearly I won't get to tell you much about story number 2. But this is the most important slide from this story. So the cool thing is that drag forces are clearly changing the lumen shape. So when we have basically a control, so this is a picture of the control, and you can see that in the control systems, basically the compression from the bottom plus the compression from the top, so drag forces from the bottom and then pushing from the top, basically generate a smushed shape like this. And in the ablated systems, instead, you basically have this much more elongated in the y direction, in the anterior posterior direction shape of the lumen. And so this is this ratio, and there's a really statistically significant difference in the experiments between the control and the ablation. And the thing that we can do is, of course, we can do the same thing in the simulations. Oh, let me sort of show you first the same thing in the simulations. So we can model sort of pushing from the top and also pulling from the exterior. And we can see that when we do a laser ablation, we really see the same shape changes that we see in the experiments in the model, like really quantitatively. So let me just say one more thing is that, of course, so you can ask, well, why when we ablate the notochord, doesn't the velocity of the Kupfer's vesicle stop entirely? And that's because we think that there are also cells at the bottom of this Kupfer's vesicle that are migrating towards the posterior. So I'll show you a little movie of that, okay? And so maybe you can see that there's sort of some migration of cell. I mean, I don't know if you can see it in this video too well, but there's definitely like lamellipodia sort of kind of fairly obvious in the cells in the bottom. And so we're exploring, we can ablate these cells, or we also have an optogenetically controlled release of a rho kinase inhibitor that also messes with the migratory ability of cells in this posterior. So we're sort of looking at that too. Okay, but I think that and this is just some simulation data, again, basically saying that if we do, so this is now sort of data from the experiment, and this is data from the simulations, and we really do get the same shape changes that you see in experiments and the simulations. It's just showing that. Okay, So I think I will not tell you, because I'm already basically at the end of my time, the story about the stratified epithelia work with, and so I apologize if some of you were really looking forward to that story. I will say, maybe I will say what we're trying to do briefly. So we're trying to understand how homeostasis is maintained in this stratified epithelia, because of course you need a balance between cell divisions and cell delamination and differentiation so that you don't get overgrowth or undergrowth of cells in a stratified epithelium. And there's several candidate mechanisms for that have been for which we have RNA-seq data, and we're trying to test those different hypotheses via these types of mechanical models in 3D. So maybe I'll just show you a quick picture of the type of things that we can do. So we can basically look at a movie where we have a cell that becomes basically committed to delaminating, and then we can sort of use the data from the both analysis of intensity of adhesion molecule expression and then also RNA-seq transcriptome data to basically say, okay, if we change the properties of this cell that we know is committed to delamination in the way that's suggested by this data, is that sufficient to allow delamination? In other words, if we just change the cell autonomous properties of this delaminating cell in a way that's consistent with experimental data, does it delaminate? And we can say, okay, what are the effects of the mechanics of the surrounding tissue, the non-cell autonomous behavior of the surrounding tissue in facilitating this delamination? For example, if the rest of the cells are more fluid-like, does that facilitate delamination of this cell or not? Or if the layer above, the suprabasal layer, if that is more solid-like or fluid-like, does that change the rate at which cells can delaminate? So that's sort of the story, and I'll just leave it there for now. Okay, so in conclusion, there are several different ways that tissues can regulate tissue fluidity, and that is important for driving morphology changes. So I guess the key thing is that since this fluid-solid transition is so important, I think in developmental processes, you want to think about which models capture those forces correctly. And the left-right organizer of zebrafish, a 3D vertex model shows that dynamic drag forces generated by fluid-like tissue surrounding the organ generate forces and drive cell shape changes. And I think the key feature for this audience is that these dynamic forces, so what I'm saying is there's a dynamic gradient in the forces that is generated by the motion of this object, and that gradient is what's causing cell shape changes. And that should be occurring, I think, in other developmental systems and could be really interesting. I didn't talk about stratified epithelia at all, I'm sorry. But I think modeling is a path for determining which mechanisms dominate in systems with multiple mechanisms where it's hard to tell what's going on. Okay, so thanks so much for your attention, and I'm happy to take questions.
Thank you very much, Lisa, for a fantastic talk. So are there questions? So I'll start with a question for you. Do you hear me?
Yes, I do.
Okay. So in the images that you show, you focused on the asymmetry that you see between the posterior and the anterior region of the Kupffer vesicle. But what is known, and what I kind of see also in your images, is that there is also a kind of left-right asymmetry between the shapes of the cells. Is it true? Do you see that? Or like was my imagination?
So, well, I don't know if it's our imagination, but we had the same thought that we see it both in experiments and simulations, but when we try to quantify it, it's never at the level of statistical significance for us.
Because,
and so
can I just add one small thing? I can see also kind of an asymmetry in your PIV analysis, in the analysis of the flows.
Yes.
Sure. Is it like, maybe it's just an example? I don't know.
No. So yes. So again, it is not at the level of statistical significance across multiple embryos. We are wondering if we had better, I mean, these experiments are exceeding, as you know, these experiments are so hard, right? But we wonder if we were able to get like 30 embryos, which is really, really challenging to also do all of this analysis, right? But maybe it would, maybe there's something there. So in one of our papers, we said, maybe there is something there. Maybe there's a left-right asymmetry in the PIV, but we're not sure. It would be cool if there was, because actually that might suggest that these stress sensors, everyone has always thought that those stress sensors that are sitting there on the anterior dorsal side that read out left-right patterning, everyone thought the only thing that matters is the fluid flow inside the lumen. But if you did have this left-right asymmetry in the mechanical properties of the tissue outside, that would also contribute to those stress sensors. And that would be an additional robust sort of, it would be a way to increase the robustness of the system if those stress sensors were both sensing stresses generated inside the lumen and outside the tissue. And that is an interesting conjecture, but one that we cannot say anything statistically significant about yet. Oh, one other thing we can do. So can I tell you a cool experiment? Because I told you we have a caged rho kinase inhibitor. So one of the things we're trying to do, and we're almost have this working well enough that it's reproducible, is basically go in and optogenetically paint one half of the embryo with this caged rho kinase inhibitor so that we really change the mechanical properties of half the embryo. And then, of course, you would expect, and actually we do have some preliminary data like this, that this organ kind of rotates because it gets more drag forces. If you change the viscosity of one half of the tissue optogenetically and leave the other, then you should actually have very asymmetric drag forces and the thing should rotate. And we actually have preliminary data like that, but nothing that I am sure is working exactly reproducibly yet. So although I don't know whether it's happening in real life, I can certainly go in and make the system do that. And I can also do it in the simulations. Yeah.
Maza, please.
Hello, it's nice talk. I have two questions. First one, a very simple one. So instead of laser ablating notocode, have you looked into a planar polarity mutant such as Bangle2? You'd expect a complete extension defect. Does alter the drag forces?
Yes. So we have looked at multiple convergent extension mutants, for example, including Bangle2. The challenge with some of those mutants, including Bangle2, is it messes up a ton of other things. And so it's hard to distinguish. So the one that's the cleanest is a ZIC3 mutant, if you're familiar. So the ZIC3 mutant is a pretty clean convergent extension mutant that doesn't appear to do much else other than alter convergent extension. And it does actually completely, yes. That mutant, because it's a clean one that only seems to really affect the convergent extension, both doesn't have the anterior posterior asymmetry in the cilia and also has significant left-right patterning downstream defects. Yes. Yeah. So it's consistent with, but I would say the challenge is that with all those sort of mutants, they're doing more things than just altering convergent extension. So we thought a notochord ablation was the most clean way to get a change in convergent extension without changing anything else.
Yeah, I agree with that. And the second question was a bit difficult to do how to experientially do it. But idea being, so if you reduce the lumen size of Cooper's vesicle from two somite to S somite, that seems to be happening spontaneously, then that would could that be secondary affect drag forces generating externally?
That's a great question. So we have a paper that appeared in eLife a few years ago now that explicitly messes with basically the ion channels and the other ways of controlling the lumen size. Because one hypothesis we had that I didn't talk about today is that changes into the lumen size are actually contributing to drag forces and also other properties in the system. So what we found is basically if you mess with the lumen size via a couple of different perturbations, that it does not impact either left-right patterning or this cell shape change And the cool thing is that you can do it a couple of different ways, because so you can do it via ion channel perturbations, but you can also do it to making the cell-cell contacts a little leaky. And so both of those things generated the same result, which give us some confidence that the change in the lumen volume is not playing a huge role here.
Okay, thank you.
Yeah.
Please, Ramya.
Can you hear me?
Yep.
That's an amazing talk. This is completely different to what I do. So please excuse my not so much knowledgeability. But is it just the size change that I was seeing, or does the KV organ also grow, or is the cells, the number of cells determined? Is it restricted? Because if you add into your model cell division within the system, how does that affect cell shape changes? And the second part was, I don't know how difficult this is, but could you do like grafting experiments where you take off this KV and put in a system where there is no drag forces? Does it still affect cell shape? In theory, like how important are these drag forces?
Yeah, okay, so I can answer the first question pretty cleanly, and I have to think a little bit about, well, actually, I don't do experiments, so like I'm really bad at answering the second question. But anyway, I can tell you what I think Jeff would say if he were here, who's my experimental collaborator. Okay, so on the first question, the cool thing about the system is really from the 2 somite stage to the 8 somite stage, we can monitor it carefully, and there's really almost no cell divisions in Kupfer's vesicle during this process of the shape changes. And you're exactly right. The lumen gets bigger, but it doesn't get bigger because the KV gets bigger, but it doesn't get bigger because more cells are being added. It actually gets bigger because the lumen is growing. And so that just sort of inflates the whole thing. And another cool feature of this system is you might think, based on what I've said, that the number of cells in the Kupfer's vesicle should be tightly regulated, but it is not. The number of cells that comprise the Kupfer's vesicle varies by like a factor of 2. It's huge. But the cool thing is we sort of, I don't have statistical significance on this, but we're sort of like, it turns out that when you change the size, the cool thing is that this mechanism I just described could be very robust to changes in size of the object, because if you change it a lot, the size of it is smaller, right? But you actually also might you're pushing on it. And so actually it might move faster or more slowly as well in a way that compensates. So I'm not saying that this is any proof of anything, but I think it's very interesting that there are these huge fluctuations in size of the organ, and yet the shape change is very robust across that, and that the mechanism that I've proposed that's at least contributing to it may help contribute to the robustness of that feature. Okay, then you asked me a second question about grafting, and I think that's really, really hard. I don't know how to do it. Because let me also say that this epithelial layer, it's not like, I think you saw, right, that it's, the edges, the sort of the effectively like kind of the basal side of them is very protrusive. So it's not like it's easy to take this thing out of its surrounding tissue. And in fact, the laser ablation in this rho kinase inhibitor set of experiments that I said we were doing, but I didn't show you data because it's all happening right now and we're still analyzing it, are the best things we think we could. That was our best way to try to perturb the system in a physical way, like, because we can't excise it and graft it and take it out, but to stop it from moving as best we can. And actually, we have done a few experiments where we've both ablated the notochord and we've put the rho kinase inhibitor in these posterior migratory cells, and we do slow it down even more. So you can kind of do a double knockout where you almost stop it entirely.
Thank you very much.
So I think that our time is over. Thank you very much, Lisa.